Hello students, welcome to Tamil online classes. So in this video, let us start with our next cell organelle, that is Golgi Complex. Very important cell organelle it is. It has many unique features as well as functions it does in our cell. Let us thoroughly study what is this Golgi Complex, who discovered it, who named it. So everything. So its structure as well as function. Let us start now. Golgi Complex. So it is one of the cell organelles. So which was the first discovered by Camillo Golgi, is the name of the scientist, in 1898. It is a cell organelle discovered by Camillo Golgi, and he reported, means it was reported for the first time, means it was discovered in the nerve cell of the barn owl, very important, as well as cat cell, right? It was first reported in, its structure was first reported in barn owl and cat. Means it was first discovered in the nerve cell of barn owl as well as cat for the first time, right? The nerve cell. You're right, very important. Okay, the nerve cell of barn owl as well as cat cell, right? And its structure, that is microscopic structure was explained thoroughly by Dalton and Felix. Dalton as well as Felix were the two scientists, the one who explained the microscopic structure of this Golgi complex, right? So this is the fact what you should know. This is the aspect regarding the discovery of these Golgi complexes. So let us start with the next occurrence of this Golgi complex. Where it occurrence. It usually occurs in mainly the secretory cells, as well as in all the eukaryotic cells. In all the eukaryotic cells it is available, and it is mainly occurring in secretory cells, right? Very important one, pancreatic cells like that. So secretory cells is mainly occurring in, and it is absent in very importantly. So matured RBCs, right? It is absent in RBCs and CO tubes. It is also absent in CO tubes and sperms of pteridophytes as well as bryophytes. It is absent in RBCs, CO tubes, as well as sperms of male gametes of bryophytes as well as pteridophytes is absent. And the shape of this Golgi complex is different. So shape of Golgi complex is different in different cells. It is different in different cells in the sense that it is variable, right? It is variable in nature, right? Based on the type of function it does and based on the type of cell it is present, right? Based on type of cell it is present and based on function, right? It's a shape that differs over here, and its number is also variable. In one of the organisms, micromonads, only one is there. In the rhizoidal cells of Tara. Tara is one of the green algae in which rhizoids are present in rhizoidal cells. So they are about 25,000 in number. 25,000 in number. And third one, in liver cell, they are about 50 in number. Means it varies from one type of cell to another type of cell, right? So the number also varying, and these Golgi complex, this Golgi complex is so present in both plant cells as well as animal cells, especially in vertebrates. So it is made up of a stake of secular tubular structures, a membranous secular structures. A stake in the sense that when you keep the coins one above the another, so that forms a stake of coins. In the same way, if the tubules are the secular disc-like structures which are arranged one above the other, it forms the what? Stake of disc-like structures. That kind of structure is seen in case of all the vertebrates. And in lower invertebrates as well as in plants, these are distributed in different areas. But in vertebrates, they are lying very nearer to the nucleus. Right? So, hence, in case of plant cell as well as invertebrates, the same Golgi complex is called as dictyosomes. Golgi complex is called as dictyosomes. In plants as well as invertebrates. In plants as well as in invertebrates, it is called as dictyosomes. Why? Because the structure is this place means it is not lying near the nucleus and it is distributed. So in the plant cells, that's why they are called as what? Dictyosomes. Okay, so this is about the introductory part regarding the Golgi complex. Then what Golgi complex is? So how to define this structure? So let us study it. So the definition is not a particular one, but I can explain or I can give one particular definition of what it is in one sentence. So it is nothing but it is a stake of a network of secular structures, membranous secular structures present very nearer to the nucleus in case of vertebrates. It is distributed in case of plants. So this is called as what? Golgi complex. Golgi complex is a network of the stake of satellite membranous structure which lies nearer to the nucleus in case of what? In case of the vertebrates. And it is distributed in case of plant cell as well as other invertebrate systems, right? So this is the Golgi complex. So it includes what? structures as if the endoplasmic reticulum, that is the cisternae are present, vesicles are present, tubules are present. These three structures together form what? This Golgi complex. Let us see the diagram first and later explain one by one. So all these components of our Golgi complex, and later we'll go through the functions of this Golgi complex. Let us start with the structure of this Golgi complex now. See here, it is made up of a state of many membranous sac-like structures over here, right? Many sac-like structures are consisting of. So let us draw the diagram of this sac-like structures over here. See here, this is how the circular structure that forms this Golgi complex. So these are usually a disc-shaped one. This kind of disc-shaped structures that we come across over here. And these are mainly the cisternaes, what we call them as. Okay. See here, this is how the Golgi complex is there. So this consisting of the state of membranous sacular structures. These are known as what? Cisternae. This is also consisting of cisternaes as well as tubules as well as vesicles. Over here, vesicles are also present. Means here also in this structure, there we come across cisternaes, vesicles as well as tubules. Let us see how the cisternaes are there in this structure. Cisternaes. So these are disc-shaped or saucer-shaped. Saucer, you know it better, right? So saucer and cup. So in which we drink the coffee, tea, etc. Right? So that kind of sac-like, membranous sac-like, saucer-shaped or disc-shaped structures these are. And these cisternaes, they bulge towards the terminal part of themselves. They little bit bulge towards their terminal part, and all these are interconnected among themselves. And these cisternaes are 4 to 8 in number in one state. Right? In one state, they are 4 to 8 in number. Right? So let us write the cisternaes. These are disc-shaped stake-like structures arranged one above the other. They are arranged one above the other. Right? So this kind of cisternaes, so they are 4 to 8 in numbers. They are 4 to 8 in number, right? So they are 4 to 8 in number, right? And such kind of cisternaes are interconnected. So these are interconnected with even the tubules as well as vesicles. They are interconnected with the tubules as well as vesicles over here. And such kind of tubular structure, that is cisternae, which have two faces over here. See here, this is the dome shaped structure. This face is called as cis face, right? So this dome shaped or convex face of the Golgi complex or the dictyosome is said to be as a cis face, or this is also called as forming face. It is also called as forming face, and this face is towards the endoplasmic reticulum over here. Why? Because the vesicles of the endoplasmic reticulum, which are so escaped from the endoplasmic reticulum, they start to mingle each other. They start to fuse, forming the tubular structure, and this tubular sac-like structure that becomes what? Cisternae, right? It is called as forming phase. The formation of the Golgi complex starts over here, hence it is said as forming phase. It has forming phase or cis phase, or it is also called as proximal phase. It is also called as proximal phase, where it is facing towards the endoplasmic reticulum. The vesicles of endoplasmic reticulum fuse to form cisternae. What it forms? It forms the cisternae over here. So already we know these are forming endomembrane system. So that's why these are all interconnected or coordinated in their structure as well as function itself. That is the same thing you must remember over here. So that is cis phase or forming phase, right? So here you are observing these are vesicles of endoplasmic reticulum. They fuse themselves forming this kind of a cisternae itself, right? So many of the cisternae are arranged over here, about 4 to 8 number of cisternae which are arranged one above the other over here. And another phases are, and this phase itself is called as a trans phase, or it is also called as mature phase, or it is also called as distal phase. Another phase is also there. What is that? So forming phase or distal phase, or it is also called as maturing phase, where these cisternae they break themselves into once again the vesicles. These cisternae break into vesicles, and these vesicles later they form lysosomes. Over here. So this is how the arrangement of this endomembrane system will be, right? So it is very important to remember. Distal or maturing phase, where cisternae break into the small vesicles forming the lysosomes. Small vesicles forming the lysosomes. This is called as a trans phase or maturing phase or distal phase also, right? So there are two phases that we come across over here in this Golgi complex. Apart from this one, next one is tubules. Let us see the tubules. So these are tubular structures that are interconnected with the cisternae towards the maturing phase. These are tubular structures present towards the mature phase, interconnected with the systems, right? So, and next one is vesicles. These are also sac-like spherical shaped structures which are associated with cisternae as well as tubules. So these are sac-like spherical structures which are interconnected with what? They are also interconnected with the cisternae itself. All these structures together form the Golgi complex itself, right? So this is the Golgi complex which is lying nearer to the what? Nucleus in case of vertebrates, but it is distributed somewhere in case of plant cells as well as invertebrates, hence it is called as dictyosome. Okay, so tubules, vesicles, tubules, already we finished. All these are the structures together form what? This Golgi complex. Let us start with the functions of Golgi complex. What are the major functions of this Golgi complex? So as you know, endoplasmic reticulum produces some of the enzymes, that is digestive enzymes, and those enzymes that are transferred to the Golgi complex vesicles, those vesicles later become what? Lysosomes. Hence, the first function of this Golgi complex is it helps in formation of lysosomes. It helps in the formation of lysosomes, right? So this is the first function of what? Golgi complex itself. And the next one, second one is very important one. It helps in the synthesis of cell wall materials. Like cellulose, hemicellulose, as well as pectin-like materials. Even it helps in formation of the cell wall matrix. That is also done over here. Okay. So and next one, the main function of this Golgi complex is, so since it is present in the secretory cells, so it helps in packaging and dispatching the materials, right? It helps in packaging and dispatch of materials. Dispatch of the material as well as packaging of the material is the major function of this Golgi complex, right? It helps in the synthesis of glycoproteins, glycolipids by one of the functions called as glycosylation. Means it helps in glycosylation. Means synthesis of glycoproteins and glycolipids. Glycolipids is also taking place over here. Apart from this one, so this also help in the synthesis of yolk granules, chymosin granules, or the type of granules it helps in? It helps in the synthesis of zymosan granules as well as yolk granules in especially oocyte. Oocyte, ovum, egg cell, you know it better, right? So in oocyte, so the yolk granules are formed over here as with the help of this Golgi complex itself. This is also one more function of what? Golgi complex. Some more functions are there. Let us see now. It also helps in the synthesis of retinal pigments. There are some pigments in the retina of the eye, right? So it helps in the synthesis of retinal pigments, and it also helps in the synthesis of the toxins of nematocyst, right? So this is also more important function of this Golgi complex. So many of the functions that we discussed. So this is the end of the Golgi complex. So and let us start with the next important cell organelle, that is lysosome now. So lysosomes are commonly known as the suicide bags of the cell. So how these suicide bags of the cells are formed, how they are, what do they contain, who discovered, what are the major functions of these lysosomes, all these are the things that we study thoroughly now itself. Let us start with the lysosomes now itself. Lysosomes. Okay, so these are single membrane bound spherical shaped organelles or the cell organelles present in majority of the animal cells especially, and few plant cells itself, right? So let us see lysosomes. These are single membrane bound cell organelles which are spherical in shape. So such kind of lysosomes, they contain acid hydrolases, or they are called as many hydrolytic enzymes. It contains many hydrolytic enzymes, and these lysosomes were first discovered by Christian D. Dewey. Is the name of the scientist, the one who discovered them for the first time, right? And these were named as lysosomes by Novikov is the name of the scientist, the one who named them, and is the name of the scientist, the one who discovered them for the first time. And so these are occurring in occurrence. So these cell organelles are mainly occurring in some of the cells. So in what type of cells these lysosomes are present? In almost all animal cells. These are present over here, and these are present in few plant cells also. In few plant cells, especially in pea seed. It is present in the PC and the root tip cells of males. They are present even in root tip cells of the males. They are present in, and so even they are present in one of the fungus, that is yeast cell also they are present, right? In plant cells, in few plant cells it is present, in almost all the animal cells it is present, and it is absent in some of the all the prokaryotes and RBCs. In matured RBC they are absent, and in almost all the prokaryotes they are absent in, right? So this is the presence or the occurrence of this cell organelle, that is lysosome. And the next one is very important one. Some of the other cells, they act as the cell organelles, so they act as the lysosomes, but they are not actually the lysosomes. Say for example, aleurone cells are there, all such kind of cells, they act as what these lysosomes itself, right? So that is the activity which is shown as if the lysosomes. Then how is the structure of this lysosome? Structure of lysosome. Lysosome, as already I have told you, it is single membrane bound spherical shaped structure which contains near about more than 40 number of hydrolytic enzymes. So these enzymes are mainly produced by the endoplasmic reticulum, and that comes to the lysosomes over here through vesicles which are formed from the Golgi complex, right? So that is very important thing to be remembered over here. So lysosomes are this way, and these lysosomes are of different types. They are of different types. So polymorphic in nature. These lysosomes are polymorphic. Wow. So based on different shapes or different structures it has. So they are, one is primary lysosome is one, secondary lysosome the second one, and residual body is third type of lysosome, and fourth one is autophagic lysosome. Are the different types of lysosomes that we come across over here based on their structure. Hence they are showing polymorphic nature or polymorphism, it is said as. So many type of lysosomes. Lysosome is single one, but it is showing different types. That's why it is said as polymorphic nature. And these lysosomes mainly consisting of 40 more type of hydrolytic enzymes I said, and those hydrolytic enzymes may be nucleases, phosphatases, sulfatases, proteases, lipases. So like that, 40 more number of hydrolytic enzymes are there present inside these. So what? So lysosomes, and these help in digestion of respective macromolecules. So nucleases, they mainly consisting of the enzymes which help in digestion of nucleic acids and phosphatases, so sulfatases, so proteases help in digestion of proteins, lipases, lipids. So like this, all these are the macromolecules which are digested with the help of these kind of what? These kind of enzymes over here, hydrolytic enzymes over here. Hence, so these lysosomes are called as suicide bags of the cells. Why? Because these help in digestion of all these materials or the macromolecules which form the entire cell itself. If the entire cell is made up of all these macromolecules which are digested by respective enzymes present in the lysosome would help in digestion of the entire cell itself. Hence, it leads to suicide of the entire cell. Hence, these bag-like structures are called as suicide bags. They are called as. Okay, so now it is very clear to us why lysosomes are suicide bags. Right? See here the cell membrane, what it is made up of? Phospholipids. Okay, so they have been digested over here. And the nucleus, what it contains? Nucleic acids mainly. They are all digested over here. So in the same way, so phosphates, lipids, so as well as carbohydrates, all these are the macromolecules which are digested by these digestive enzymes, in the sense that whole cell is digested by these enzymes which are present inside this lysosome. Hence, lysosome is said to be as suicide bags of the cell. Now, let us start with the different types of lysosomes which show polymorphism. I said, so the different types of lysosomes and their respective functions. So let us see now. Let's come to the types of lysosomes now. First one is primary lysosome. So these are very important to remember. See here, primary lysosome. So I shall discuss this one. So one of the cells that contains the nucleus. Near the nucleus there is what? What is this? Golgi complex. Golgi complex is there. See here are some vascles. These vascles are pinched off this cisternae. It comes out of the cisternae, and these vesicles that contain the hydrolytic enzymes called as primary lysosomes. Primary lysosomes, these are newly pinched off vesicles with hydrolytic enzymes, right? With hydrolytic enzymes called primary lysosomes. Now it is very clear that from this cisternae of the Golgi complex, some of the vesicles are pinched off, and these vesicles, if they contain hydrolytic enzymes, then they are called as primary lysosomes. It is very clear to you. And second one, secondary lysosome. So this is more important one. This is more important one. Why it is more important one is because, so you know phagosome, how it is formed, you know it better. So phagosome is formed as a result of endocytosis, right? So endocytosis leads to formation of phagosome. Already I have told you, I had shown you, so in the last class. So once again I will show. So this cell has membrane, and this membrane has so near to this there is a food vacuole, or there is a food material which is lying near this membrane itself. So what happens over here? This membrane that extend outwardly around this food vacuole or food material, and this food material is engulfed, and that forms the phagosome. This is the food and this is the vacuole, right? And this forms the phagosome. These two together form phagosome. Now what is phagosome? You can see here there is a newly pinched off lysosome is there here, and this lysosome fuses with the phagosome. Fusion of lysosome with the phagosome. These entirely called as what? Secondary lysosome. Secondary lysosome. What do you mean by secondary lysosome now? It is a type of lysosome in which phagosome fuses with the lysosome. Phagosome fuses with the lysosome, forming what? Secondary lysosome. Secondary lysosome. This is about the secondary lysosome. And third one is very important one. What is the third type of lysosome I have written over here? So that is autolysosome as well as autophagic lysosome. It is also called as autophagic lysosome is one type, and so next one is residual body is another type. So let us discuss the residual body first and later autophagic lysosome later. So residual body. What is this residual body? Simply residual in the sense that the waste body. Alright? See here, what happened over here? Newly pinched off vesicle that fuses with the phagosome. Now it becomes what? Secondary lysosome. In this secondary lysosome, what happens? All these hydrolytic enzymes digest this food material in the phagosome. Is it very clear? Now let us see. This is the phagosome and the lysosome. This is the food, and these are the hydrolytic enzymes. What happens? Hydrolytic enzymes will digest the entire food material. They digest the entire food material. After the digestion, the digested food is removed from this lysosome. The useful digested food material is released into the cytoplasm. It is released into the cytoplasm. What remains inside this one? So there remains the undigested food. What remains inside it? The undigested food. Undigested food present inside this vacuole, and this undigested food containing vacuole itself is called as what? Residual body, right? And this residual body is removed from the cell by exocytosis. The waste products have been removed out of the body of the cell by exocytosis. What are removed over here? Waste products are removed over here in the form of the vesicle. That vesicle containing the waste products or the undigested food called as residual bodies. It is third type of lysosome. lysosome which contains the waste or undigested food. And fourth type is, what is the fourth type? Very interesting one. Autolysosome, or it is also called as autophagic lysosome. Why it is called so? Why it is called so is because, see here, some of the primary lysosomes, they gather themselves around the worn out cell organelles or worn out the ingredients of the cell. Whatever the cell organelle it is, so around that one, all the primary lysosomes, they gather themselves, they fuse themselves around that one, forming a structure inside which what is present, one cell organelle is there. All the primary lysosomes, they release the hydrolytic enzymes over the worn out cell organelle. So waste cell organelle, the cell organelle which has stopped, non-functional one. So in that, so all the hydrolytic enzymes are released. So that what happens? Digestion occurs. Okay. So there, killing of the cell organelle, that is worn out cell organelle, that takes place over here. That type of lysosome is called as autophagic lysosome. See here, let us see with the diagram over here. See here, this is one mitochondria. This is one mitochondria, right? Imagine. And here are some lysosomes, primary lysosomes. They fuse around this mitochondria. So they contain one hydrolytic enzymes are present. Hydrolytic enzymes are present over here. So now what happens? All of them release the hydrolytic enzymes on this mitochondria. What happens here? The digestion of the entire mitochondria that takes place. And this kind of lysosome itself called as autophagic lysosome. Autophagic lysosome, or it is also called as autolysosome. Right? So it digests all the entire mitochondria, and later it becomes one type of lysosome that is called as autophagic lysosome, that is autolysosome. It is also called as. These are different kinds of lysosomes that we come across, hence these lysosomes are polymorphic in nature. Poly means many, morphic in the sense that, so morphological features, that is externally looking to be, right? So these are different types of lysosomes that we discussed. So now the functions of these lysosomes, let us see now. The functions of lysosomes mainly consisting of the digestion, especially the intracellular digestion it includes. See here, so different types of functions these lysosomes does. They are very interesting one by one. Let us look in, right? The first one, the first function of these lysosomes. See here, so these lysosomes will help in digestion of the intracellular debris of the intracellular waste products, right? So simultaneously the engulfed extracellular pathogens or extracellular other materials are also digested over here, right? So these help in digestion of intracellular waste and even engulfed extracellular pathogens or other materials. Right? So this is the first function of this lysosomes. Second one is they help in the self-destruction of the cells. Right? See here, some of the worn out cells are there. So those worn out cells, so they have been digested by the own lysosomes itself. Right? So the lysosomes help in self destruction of the cells. What kind of cells? Worn out cells. So this leads to the self destruction. Hence these are called as suicide bags of the cells. What they are called as? Suicide bags of the cells. When the digestion of all the entire cell organelles or all other things which are worn out one, so the products which are formed as a result of the digestion, so those products are utilized for construction of the new cell organelles over here. So hence these are also acting as recycling units. Lysosomes act as recycling units. Why? Because the digested products are reutilized to construct the new cell organelles. Hence these are called as recycling units. So these are some functions. Next one is so they also help in histolysis of the larval stage during metamorphosis. See here, in larval stage, how the larva of the tadpole of the frog is there, you know it better. So after that, the next stage is adult stage. In adult stage, it has a limbs, but in larval stage, it doesn't have any limbs. Right? So there are some histones or the tissues, they have been digested over here. And the products of the digestion are utilized in construction of different parts over here. That's why, so it helps in means lysosomes help in histolysis. Histose means tissues, lysis means breakdown. Histolysis during metamorphosis. Already I have told you what is metamorphosis. All the changes from the larval stage up to the adult stage, all those changes are said to be as morphological changes called as metamorphosis. Right? So this is also one more function. And ecrosome. Ecrosome is the largest lysosome. Is the largest lysosome present at the tip of the sperm. See here, present at the tip of the sperm, there is the largest lysosome is present. It is called as ecrosome. So this ecrosome help in digestion of the cell wall or the wall of the egg cell. See here, this is egg cell, this is the nucleus, here comes the sperm. Right? This is the sperm. So sperm, when it touches the wall of this egg cell, so there the digestion of this wall material has been taken place by the tip of the sperm called as ecrosome itself. The tip of the sperm consisting of the acrosome, it is the largest lysosome that helps in digestion of the wall of the egg cell. This is also one more function of this lysosome. And the aged cells, worn out cells, right? So those degrading cells, they have been removed by this lysosome. Aged cells, dead cells, or worn out cells are removed by a lysosome. Say for example, regression of uterus after delivery. During delivery, many of the cells they die, right? After the delivery, the regression of the uterus that take place is done by, so there the dead cells have been removed by these lysosomes only. Okay, so this is also one more function of lysosome, right? And the next one is very important one. Some of the WBCs as well as macrophages, they act as what? The defense system of the body, right? Why do they act so? They act as the phagocytes. So phagosome you came to know. Okay, so let us see many of the WBCs as well as some of the macrophages help in defense system of the body. Why? Because they fight against the pathogens. They fight against the pathogens. After this one, what happens? The lysosomes digest those pathogens, right? So those pathogens. So these are some of the functions of these lysosomes. Okay, by this we finished the two cell organelles. One is endoplasmic reticulum, second one is Golgi complex, and third one what we finished now itself is the lysosomes. Next one is vacuoles. We'll discuss in the next video. Thank you.